I'm Braniste and I postdoc at the Neurogastroenterology and Nutrition Unit at the INRA Institute in Toulouse, France. I was asked by the DW committee to present my abstract showing that the xenoestrogen Bisphenol A induces the premature closure of the intestinal epithelial barrier in neonate female rats. It is well known that intestinal barrier fulfills multiple complex roles by providing a barrier from the exterior and a platform for antigen presentation and immune stimulation. During the perinatal period, which is extremely important for the development and maturation of the gastrointestinal tract, the high intestinal permeability from birth assures the passage of small quantities of antigens essential for the maturation and the development of intestinal immune system, while the exposure of the adult female rats to the environmental xenoestrogen Bisphenol A, widely used in food packaging industry, improves the intestinal epithelial barrier through estrogen receptor beta decrease of the intestinal paracellular permeability. In contrast, the perinatal exposure to the xenoestrogen Bisphenol A increases the susceptibility for developing colonic inflammation in female offspring in adulthood.
Based on these observations, the aim of our study was to evaluate the impact of the perinatal exposure to low doses of bisphenol A on the intestinal permeability, tight junction protein expression, and intestinal transport of the bacterial products during the postnatal gut maturation. First, our study revealed elevated intestinal permeability from birth, which decreases progressively during childhood to levels similar in adults, a process called maturational gut closure. And this process correlates with low mRNA expression levels of tight junction protein genes, occludin and junctional adhesion molecule A, that control the paracellular spaces. As we can see in these graphs, a twofold lower expression during the first 10 days of life compared to adult colon. Furthermore, we have also noticed high levels of estrogen receptor beta mRNA expression in the colon from the fetal stages, which decreases progressively during the first 10 days of life. Despite the absence of the circulating estrogens during the perinatal period, these estrogen receptor beta are functional, as we have shown that administration from birth of subcutaneous injection of 5 microgram per kilogram per day of estradiol benzoate in female pups induces 35% decrease of the intestinal permeability compared to controls. Thereafter, female rats were exposed to oral bisphenol A from the gestational day 15 till birth, and then during lactation at 50 microgram per kilogram per day, which corresponds to the tolerable daily intake or the lower dose of 5 microgram per kilogram per day. And in the offspring females, we measured the intestinal permeability, tight junction protein expression, and the transepithelial transport of the lipopolysaccharide, which is the major cell wall component of the gram-negative bacteria. So, the both low doses of bisphenol A induce a significant decrease of the intestinal permeability in juvenile female rats, minus 33% for the TDI and minus 42% for the lower dose compared to controls. The decrease is almost similar to those seen after direct administration of estradiol in pups and demonstrates the great sensitivity of the intestine in the perinatal period to environmental xenoestrogen bisphenol A. The reduce of the intestinal permeability by the lowest dose of bisphenol A, 5 microgram per kilogram per day, is due to significant overexpression of the estrogen-sensitive tight junction proteins, occludin and junctional adhesion molecule A, that control the paracellular spaces. Moreover, the decreased intestinal permeability by bisphenol A is maintained during the whole developmental period, suggesting a premature gut closure, and this process is associated with reduced lumen to mucosa transport of the lipopolysaccharide in basal conditions, necessary for the development of intestinal immune system. In conclusion, our study shows for the first time that low doses of bisphenol A, in particularly the 5 microgram per kilogram per day, a dose 10 times below the tolerable daily intake, decreases the basal intestinal permeability in the juvenile female rats and limits lumen to mucosa crosstalk between bacterial product and mucosal immune cells, which can result in impairment of the intestinal immune system maturation associated with gut barrier dysfunction later in life. Thank you for your attention.